7, thank you. Thank you, Eve. And thanks for the invite to the meeting. It's very timely and a very mixed crowd of scientists, clinicians, and lay people. And it's useful to have everyone in the same room. So let me talk a little bit about the challenges in drug discovery and start with what do we know? Well, we know there's a lot of drugs to treat pain that are available. You can go to over the counter, or you can go to your doctor and get a prescription. And the count that I came up with was 552 different drugs to treat different types of pain. And if you look at what they're treating, you've got 216 alone that are prescribed or available for back pain. You've got 272 for various types of headaches, from mild headaches to serious headaches. And so what's the problem? We got all these drugs. They're already available. Well, it turns out that that is the problem, in the sense that if you look at what those drugs are doing, they're not actually targeting very many different parts of the pain pathway. Dr. Zamponi and some of the other speakers have already shown you, Gilles, that there's central parts of the pain pathway in the brain, the spinal cord. There's peripheral, nerves, muscle. And if you look at the targets, the actual molecular targets that these pain drugs, these 552 drugs are targeting, it's not actually that many. You've got, for mild pain, you're basically targeting with the NSAIDs a family of a couple of genes called COX-1, COX-2. When you get into the mild or moderate pain, you've got more stronger NSAIDs, more specific NSAIDs. And then you get into the opioids. You've got some epileptics and antidepressants. These are also very limited targets that are being targeted with these hundreds of drugs. And then in the severe pain category, you've basically got opioids and gabapentinoids. And so gabapentinoids target a very specific molecular target. Opioids target very specific molecular targets. And so what's the problem when you look at the physiology? The physiology is clearly way more complex than a half a dozen or a dozen targets that these 552 drugs are going after. And so what is needed is we need to target different aspects of the complexity of human pain. We need, obviously, to, as mentioned, there's over a couple hundred kinds of neuropathic pain alone. There's peripheral pains, there's central pain, there's acute pain, there's chronic pain. We need to get away from opioids, obviously, for the properties, adverse effects, and death, which is the most adverse effect you can have, clearly. And then we need actually biomarkers. It's not really come up at this meeting. We need to actually have markers for following whether the drug is working or whether the disease is progressing, and how the disease is progressing. And none of the drugs around, or there are no really good biomarkers. We have the pain scores that people are subject to or take these tests, but those are very subjective. And so there's no random, so not random, there's no non-biased pain markers that people take. We need to have something such as MRI or something similar to gauge how drugs are actually working. So what is the drug development process? Just to remind you, it starts off here in either an academia or in a company, and there's some target identified, an opioid receptor or what have you, way back when, and then that target is turned into a screen, and then it's validated in animal models. And this can take years, 3 to 5 years on this scheme, and then it goes into safety and toxicology. Is it going to be safe in humans? And then there's a long period where it's tested in human clinical trials, 1, 2, 3, and then it's put up for market approval either in Canada, Europe, the US, what have you, Japan. And so this is a long 10 to 12-year process. And part of the problem is this is expensive. I've gone through this a couple of times. It's probably, the real number is probably around $80 to $100 million per target that you start with here, and per drug that comes out of this to go through this. So $80 to $100 million. And the time, it takes a long time. Pharmaceutical companies, biotech companies, especially small biotech companies, don't have a lot of money, and they don't have a lot of time before they have to return investment or money to their investors. And so this is a bottleneck, but right here is a huge bottleneck right here, as I'm going to come to here, in the sense that if you look at simply the peripheral potential targets for pain, that have been identified in animals, models, is there are literally dozens and dozens of potential single molecular target. Oops, sorry. Oops, how do I go backwards? The red one? Oops, go backwards. There we go. So if you look, there's dozens of targets that are being explored in academia, in industry, and they all target individual molecules that are in some way involved in either sensing pain, transmitting that pain through to the spinal cord in the brain or back, or modifying that pain signal. And the last time I counted the number that industry were working on, biotech and pharma, there were 64 different individual molecular targets. And so this is a problem. You can't pick each one of those, spend $100 million on each one, and go through that 10 or 12 process in the reality of the world we live in with regard to drug development. You also, I'll skip that. You also have the issue that there's not a lot of evidence in humans that a single target's going to affect very many kinds of pain. Other than the opioids, and Gerald Zamponi mentioned a particular target that humans have defective, a particular sodium channel. Human pain is much more complex than that, as we all know, and a single gene is not going to do it. So targeting a single gene is probably not going to affect very many types of these 200 kinds of neuropathic pain, and it's largely probably going to fail because it's not going to be effective in that grand scheme of how all of those different proteins and factors are interacting with each other to cause that pain pathway forward and backward in the human system. So it also, the single target also ignores the complexities of biological systems. None of those single targets acts by itself. There's circuitry in the spinal cord and the brain, which is positive and negative, it feeds back. And there's tremendous redundancy built into your nervous system. Otherwise, development and surviving would be very difficult simply by affecting one target at a time. And so, let me go forward here. The other issue is that what scientists use in that very early stage of drug discovery is they use rats and mice. And rats are not humans, and mice are not humans. They're convenient, they're cost-effective, and they're career-effective in the sense that you can work with them at very early ages, a few months, a few weeks old to a couple of months. But we clearly know these are not very translatable models to humans. So the whole bottleneck begins right at the very beginning of the process. And if you look at a gabapentinoid, for example, they work in 20, 30 minutes. And all of the tests that are done in research, those drugs work in 20 to 30 minutes in all the animal models, but in humans it can take 2 or 3 weeks to have an effect. So there's something clearly wrong. Or a bottleneck here. And then there's all kinds of, the models that are actually used don't really relate to the disease indications in humans. We inject drugs under the skin to cause inflammation. We inject drugs into joints to cause osteoarthritis. We actually surgically modify nerves directly. We inject drugs to mimic diabetic neuropathy. So there's a huge problem both in having single targets and in following up these single targets in animal models. And let me just skip to that. So what is needed is we need to get away from the reductionist single target model. We need to expand that into more physiological models, and we need some incentives for scientists and clinicians and industry to actually work together into networks. We need financial collaborators and incentives across Canada and the US and here and within industry. And specifically within Canada, we need to have CIHR incentivize researchers to perform translational research. This is not really a major priority of CIHR. It's something that's definitely needed to actually get back to the very basics of that bottleneck and address some of those issues. Thank you.